Okay, so we're going to talk about lipids. And in this video, we're going to talk about the different classes of lipids and their functions. And then in the next video, we'll talk about how we use different types of lipids for energy and some of the metabolic processes that they're involved in. So lipids are involved in biological membranes. They're used for an energy source. They're used for cellular communication. And a lot of other different lipids have a lot of other roles, but these are probably three of the major roles. We're going to go through the classes of lipids, and we will see some of the specific roles of each of the classes. So something to remember about lipids, remember when we talked about the 4 classes of macromolecules, lipids are not polymeric. So they didn't really fit into that table that we made at the beginning of the quarter where it showed the monomer form, the polymer form, and the linkage between them. They have a big structural variety. There can be lots of different types of shapes and branches, ring structures within lipids. So this is different than carbohydrates, proteins, and nucleic acids, where those are largely polymeric. One thing that is similar for all lipids is that they are non-polar, or they at least have a non-polar component. Meaning that they are not soluble in water. They are insoluble, which makes them good for biological membranes. So lipids are involved in forming what's called a lipid bilayer. So lipid bilayers are what make up biological membranes. And it's called a bilayer because it's made up of 2 layers. So lipids have 2 parts. They have up here, which these circles are. These are what's called the polar head group. And then they have these non-polar tails. And depending on what type of lipid these are, this would look different. But they form a lipid bilayer. So this would be one layer of lipids together. And then there would be a bilayer. And what happens is the polar head groups face the aqueous water-containing sides of the environment, and then the non-polar tails face each other. So most membranes are called lipid bilayers because they are made up like this. The first and kind of the simplest class of lipids is what's called a fatty acid. So these in the lipid bilayer would be examples of fatty acids. So here this would be again polar head group, non-polar tail. So again, this would be hydrophilic. This would be hydrophobic. These can exist in various lengths. These tails are usually 14 to 16 carbons long, but they can vary, and we'll look at some of those. Fatty acid synthesis adds carbon units in groups of 2. So these naturally occurring fatty acids are all going to be even, have an even number of carbons because they are put together in groups of 2. So at the end here, this hydrophilic polar head group is made up of a carboxyl group. So I'll just draw this maybe over here. And at physiological pHs, this carboxylic acid would have lost a hydrogen. And then here, you would have this hydrocarbon chain. And that would be made up of carbons and hydrogens, depending on however long it is. This is an example of a saturated fatty acid. It's saturated because there is no kinks, and if we were to draw the complete structure, you would see that each of these carbons is saturated with hydrogen. So when you hear saturated versus unsaturated, it has to do with the saturation of carbon with hydrogen. So if I was to draw you an unsaturated fatty acid chain, there would be a kink in it. And a double bond. And this would cause an angle. I didn't draw it very good. It looks pretty straight. But it would actually come off at like a bigger kink. So this is unsaturated. So unsaturated fatty acids are not saturated with hydrogens because the hydrogens have been lost here. When the hydrogens are lost, that forms the double bond between these two carbons. This causes a kink in the tail, and this is important for shape and what we call fluidity in the membrane. So if you think about if you had a membrane that was made up of all saturated fatty acids, they're all straight, they're going to pack together very tightly. This would make it more rigid. If you have a membrane and it has unsaturated fat in it, unsaturated fatty acids, they're going to be more fluid and they're going to pack less tightly together. So this is why saturated fats are so bad for us in our diets, because saturated fats are more likely to form solids inside our bodies, like in our arteries. Saturated fats are more likely to form plaques and arteries in our, plaques and blockages in our arteries. Unsaturated fats are less likely. So this is why you'll hear, that saturated fat is bad. A lot of animal fats are saturated, and a lot of vegetable or fruit fats are unsaturated. So that's where this comes from. And there's lots of pictures in your textbook and in the slides of cartoons of saturated versus unsaturated fatty acids and what that makes the lipid bilayer look like. So all of these lipids having a polar head group and a non-polar tail group, that makes these membranes what we call amphipathic. Amphi means both, and it can exist in both environments, polar and non-polar. And you see in a membrane situation, the polar head groups will orient themselves towards the aqueous environment. The non-polar tail groups will kind of hide from the water and face each other. If there is, back to this one, if there is a double bond, most double bonds in nature are cis. Trans double bonds actually don't make a kink, and they actually pack together tighter than saturated fats. That's why trans fats are very unhealthy for you. And they also don't exist in nature very often. They mostly have been artificially manufactured. So this is kind of the first class of lipids that we'll talk about, and we will talk about how we store fats as fatty acids and how we use the next molecule that we're going to talk about is called a triacyl glycerol, and it's going to have 3 of these fatty acid chains. Okay, so here's the next class. It's called a triacyl glycerol. You might also hear it called a triglyceride. Tri means 3, acyl are these chains, and a glycerol. So a triacyl glycerol looks kind of like this. This would be your glycerol molecule. And it's going to have 3 fatty acid chains coming off of it. And they could be saturated or unsaturated. There's all kinds of different forms of these. But it's a tri-3-acyl glycerol. These are fats and oils in plants and animals. They are water insoluble, and this is a good way to store energy in animals. Fats and oils are a mixture of triacyl glycerols and fatty acids. In science, in a biology or a chemistry class, you would hear these called triacyl glycerols. In medicine, you would have it called a triglyceride. So if they're testing your blood levels and they're testing your triglycerides, this is what they're testing. So we need to store fat for 3 major reasons. One is we use fat for energy production. We actually use fat for about 1/3 of our energy consumption. Also, there are some organs that prefer fatty acids as their fuel type, so we're going to see that in a little bit too. We also use fat for heat production. So there's specialized cells in tissue called brown fat. So warm-blooded animals have this brown fat. Animals that hibernate or that live in colder climates, they will have higher or lower concentrations of what's called brown fat as compared to what we call white fat. We also use fat for insulation. We use it for the cold, but we also use it in different parts of our body, kind of as like shock absorbers. So in the bottoms of our feet, between some of our joints, we use fats for lots of different things. We store these fats, these triacyl glycerols, they're stored in a special type of cell that's only for storing fat called adipocytes. So these fat storing cells store triglycerols or triglycerides, and this is what we would use in times of starvation. This usually we don't use the fat stored in our adipocytes until after the glycogen and other sources of energy have been used. We'll talk about this a little bit more later when we talk about beta oxidation of fatty acids and how we metabolize fats. Let's see. We've talked about membranes a little bit. Membranes are made up of all types of different lipids. Okay, the next class of lipids are called Glycerophospholipid. Glycero means there's still a glycerol. Phospho means there's a phosphate group. Lipid. So these can vary. Usually what a glycerophospholipid looks like is you have the glycerol. Instead of having 3 fatty acid chains, you usually have 2 fatty acid chains, and then you'll usually have a phosphate group on this side. Because remember, this phosphate group is charged. Charged things are soluble in water. This glycerol is also soluble in water. So this phosphate group might get synthesized on this side, and then it's going to flip over because it's amphipathic. This part of the molecule up here with the glycerol and the phosphate is going to be polar. This part of the molecule down here with the hydrocarbon is going to be nonpolar. So you might see these drawn either way. You might see the phosphate group on the same side as the fatty acid chains, or you might see it flipped over. These are, there are many, many different variations of these glycerophospholipids, and we are not going to talk about all of the specifics. Any of you in neurobiology will talk a lot about these types of lipids in signaling in our brain and in our nerves. We're not going to talk a lot about those today. Another class of lipids that we can talk about are called sphingolipids. So sphingolipids are sometimes called ceramides, and they include a molecule called sphingosine and a fatty acid chain, and then some other molecule there, a ceramide, phosphatidylcholine, glucose. There's all kinds of different variations to this. These are the most common. They're the most common lipid. They're the most common lipid in, let's not say that. They're the most common in our brain and our nerve. These constitute the membranes around the myelin sheath. They insulate our nerves and are really important in nerve impulses and found in a high concentration in our brain. And again, there's lots of good pictures of these in your textbook and in the PowerPoint slide. So cerebrosides are important Glyco Sphingolipids. They're a sphingolipid with a glyco group. A ganglioside. These are examples of sphingolipids. They're the most complex. And anybody who's taken any neurobiology courses will have learned a lot about these. But we're not going to spend a whole lot of time on them because they're not something that are heavily involved in metabolic processes, and that's where we're really going to function. The last class of lipids that we're going to talk about are steroids. So steroids are a type of lipid. They usually have many rings. Instead of having groups and then long fatty acid chains, they usually are what's called heterocyclic, meaning they're made up of multiple rings. On slide number 29 in your lecture, there's a picture of cholesterol. And cholesterol has 4 rings, 3 6-carbon rings and 1 5-carbon rings. And then it has some different functional groups coming off those rings. A lot of hormones are steroids, testosterone, estrogen, progesterone. A lot of those are steroids. Steroids are also important in biological membranes. It doesn't look like there's no pictures here of steroids or cholesterol in our lipid bilayers, but lipid bilayers actually require cholesterol especially to maintain fluid mobility. So it actually, there's a kind of a perfect ratio for which cholesterol makes the fluidity of a membrane just right. Too little cholesterol decreases the fluidity. Too much cholesterol decreases the fluidity. But cholesterol is important in maintaining membrane fluidity. Because we want this lipid bilayer to be fluid. It's kind of like you could think of if you had a pool and it was full of balloons or full of balls, and nothing was moving on the surface of that pool. Everything would touch each other, but if there were some waves in the pool, everything would kind of move together. So you want it to be fluid. Transport across membranes is not something that we spend a whole lot of time on in this class, but we need membranes to have some fluidity for transport. Also, they need to have enough rigidity to maintain the structure of the cell. So making sure that a lipid bilayer has just enough fluidity and rigidity is an important role of different types of lipids. So there is a slide number 38 in your lecture, and it shows what's called the fluid mosaic model of biological membranes. It shows this lipid bilayer with a cross section. It shows proteins that are spanning the membrane. It shows cholesterols that are being embedded in the membrane, and it shows things on each side of the membrane. This is actually a lot more complicated than just this cartoon that we're looking at, but the majority of the structural component of lipid bilayers, which every cell in our body has, comes from lipids.